Okay, so today I will talk a little bit about our new work on the impact of pathogen quiescent on epidemiology models, and also I'll try to briefly mention some methods we developed to look at the genome-wide polymorphism data to understand the nature of host-parasite interaction. So let's start. Yeah, as we all know, species interactions are the building block of any sustainable ecosystem. So if you look around, you see that all species interact with many other species. And these interactions can be broadly classified into three different classes based on their impact on each partner. So for example, if both partners are getting a benefit out of this interaction, we call it as mutualistic, like a bee and a plant interacting. So both are getting some benefit in this case. So that's a mutualistic type of interaction. You could have another class of interaction, which we call it as a commensal type of interaction, where one species is getting an advantage, but other is mostly unaffected. And the last class is the antagonistic type of interaction, where one species is getting a disadvantage and the other is getting an advantage. So basically, we are mostly focusing on only this class of interaction, where this one species, say the parasite or a pathogen, getting a fitness increase at the expense of the host fitness. And these interaction, of course, you can imagine that there is this negative feedback, negative selection happening on each species. Like when an infection disease spread in a population, it will create some sort of population dynamics in both species. So the population size of the host and the pathogen get affected when an infection spread in the population. Moreover, we know that there are some host pathogen pairs which kind of goes on for a really long time, and that will result in some sort of coevolution. So these two species will interact with each other if in a very long time scale, in the evolutionary time scale. So it will impact the genome evolution of both species as well. We want to look at both these scenarios. So we wanted to look at the population level changes, and also we want to look at the genomic level changes, like how the genome of both these species will change because of this interaction. So basically the questions are, we wanted to understand how the population size of host and pathogen changes, mainly for a model where we allow the pathogen to have dormancy or quiescent properties. And also we will try to identify the genomic regions which kind of drives this coevolutionary interaction. Okay, so let's start from the most simple model setting scenario. We have a host, a plant or an animal, whatever, and a parasite. And the parasite attacked the host, and host tried to develop defense mechanisms against it. So this is basically the interaction we are interested in. And this creates some infection in the population. So I can categorize my population into two boxes now. So I have the healthy ones and the infected ones. So I call the healthy ones as the, as the susceptible individuals and put it in this class. And I have the infected ones as I and then put it in this class. And then I can go from this healthy individual to the infected class if they get infected. And this can be described, this number change in these two classes can be described by these two equations, which look simple, nice, and we know almost everything about the symbol system, where it has two possible stationary state. One is a disease-free equilibrium, where you eliminate the pathogen from the population, so your I class get empty and everybody is in the susceptible class and no infections anymore. Or you could also have an endemic equilibrium where you maintain a fraction of the population in the infected state. So this is what we have for this model. We want to go one step ahead to consider a system where you have still one host, but two pathogen strains. So this is quite normal in nature, because even with corona, you know that there is multiple strains, right? So there's all pathogens kind of shows many diseases, there is coexistence of multiple strains or multiple strains evolving at different time points. And we also wanted to go one more step ahead to allow this pathogen to go to this dormant state once in a while. So you have this active pathogen, it it creates the infection. And once in a while, it could go to a quiescent or sleeping box where it is inactive for a bit, and then it can come back to the active state after some time. So this is basically the model we want to work with. And most of this work is done by Usman, who is a PhD student with Aurelian and Johannes. And yeah, I'm also involved in it briefly. So this is the model setting. And then, of course, we wanted to put all the parameters of this model, which is basically the rates of moving from each of these boxes to the other boxes. So the first one, this green arrows can be considered as a constant birth term, which kind of give the inflow to the healthy class, which can be birth or migration of healthy individuals to the population at a constant rate. And then you have the natural death, which is all these red arrows going from all these boxes. All individual has some probability to die at some time point. So you have the natural death term of outflow from all these classes. And this is this gamma is basically virulence, which is added to the death term in the infected classes, which is the disease-induced death. So if you are infected and if you have disease symptoms, you might have some more chance of death, like because of the disease symptoms. So this will give a higher death rate for the infected individuals. It's not there for the quiescent ones because they assume to not show any disease symptoms. And you have the transmission rate, which kind of, when a healthy individual encounter an infected individual, the infectant get transmitted and they move from the healthy to the infected compartment. And you have recovery, which take the infected ones back to the healthy compartment. And then this is all the normal model setting for the infected and the healthy class. And there is also switching to this dormant or quiescent state. So there's a rate rho with which the individuals infected by pathogen 1 could go to a quiescent state. So basically, the pathogen 1 could decide to sleep for a while and go to this quiescent state at rate rho, and they can wake up at rate zeta. So these are the parameters of a model. And then we can, of course, write down the differential equation for the number change of each of these classes based on these parameters. Okay, so this is our simple system. But when we kind of look at the system carefully, we see that it has 3 possible equilibrium state. One is a disease-free equilibrium state, which is of course not very interesting. It says that all these compartments stay empty, all the infection vanishes from the population. And 2 other possible equilibrium states are the boundary equilibrium, where you can only preserve one pathogen, like parasite 1 can survive, but 2 get eliminated, or parasite 2 can survive, 1 get eliminated. But we are more interested in having a polymorphic equilibrium state. So is it possible to keep both the pathogen in the system? Mathematically, it can be done by adding a constant here. But biologically, is it realistic? It's not completely unrealistic if you assume some sort of migration of infected individuals into the population as well. So it's some sort of forcefully keeping the individuals, but so this biologically, this constant can be interpreted as a constant level of migration of infected individuals to the population. If I have this epsilons of constant migration rate to my population, then you can actually preserve both the pathogen in the system. Then we can actually write down, so then we wanted to evaluate the stability of this polymorphic equilibrium. For that, we follow the standard procedure of writing down the Jacobian of the system at this equilibrium point, and try to so the standard procedure is, of course, get explicitly calculating the eigenvalues, but it's hard to calculate the eigenvalues for such high dimensional systems. So basically we follow the standard procedure of looking at other theorems which could give us some information about the sign of eigenvalues. And yeah, I mean, Osman and with the help of Johannes and Aurelian managed to get this right. So they reformulated this root house criteria for this particular setting and tried to prove that this and they managed to prove that this polymorphic equilibrium we have is locally asymptotically stable. So we could actually prove that this with this condition, that if there is migration, you have a polymorphic equilibrium in the system, and that is stable as well. Okay, so now we have this polymorphic equilibrium existing in the system. So now we wanted to see how stochasticity will influence the system. So for that, we follow this, we write down the transition rates for all states, and then we follow this Gillespie algorithm to introduce stochasticity into each of these transmission rates. And then we get this master equation, which of course look a little bit scary. And then so this equation, this equation is describes the system with stochasticity. And now we go ahead and try to write down so to understand the dynamics of the system, you have to translate it to the Fokker-Planck setting. So we managed to get the Fokker-Planck equation, which is also quite big and difficult to manage. But we can actually linearize this multivariate Fokker-Planck equation around the stationary state of the deterministic system, and that will give us this somewhat nice looking Fokker-Planck equation, which is linear also. And there, this y is basically the variable which captures the number changes in each of our classes. And these matrices are this, so basically this n and m are constant matrices, which we obtain from the linear transformation of the Fokker-Planck equation. And this particular equation has a normal distribution as the solution, where we somehow know all the parameters except this one, which is the covariance matrix, which is also defined in terms of n and m. So we have, so we proved the existence of this polymorphic equilibrium, and then we also managed to simplify the Fokker-Planck equation and get a normal solution for the distribution of individuals in each of our classes. So now the next step is, of course, to validate this what we got. So we have this normal distribution. So does it really capture the stochastic system? Or what we get in the stochastic system. So it seems like it's working pretty well. So so here Usman did simulations on the Gillespie model we have, and this histogram is the distribution, or the distribution we got from the Gillespie simulations. And then he also plotted an approximation of this histogram as a green line, which is basically the normal approximation of this Gillespie model. And then the red line is basically what we get from our solution of this linear Fokker-Planck equation, which is this normal distribution, which actually matches quite well. So here, so this kind of says that this linearization technique somewhat works well, and we could actually describe the population, the distribution of population in each of our classes using this, this result we get from here. Okay, so this is what we have, and then we wanted to analyze the system with what is the effect of dormancy in the system. So basically, we look at two cases. One is a symmetric case, which means that both strains are identical. So you could do whatever you want. So first, we will try to make all these rates of strain 1 and strain 2 same. So then you effectively have two equivalent strains, so they are identical, so there is no competition between strain, and we could only look at the impact of dormancy in the system, like when there is no competition between strains. And then we also consider a case where one strain has the ability to go to quiescent and other does not. So then you have difference between these two strains. And then there is competition between these two strains. So then we also wanted to look at what is the effect of the dormancy in this particular scenario when you have asymmetric rates, and how does the infection and the stochasticity in the system looks like when you have this competition between strains. Okay, so first is the symmetry case. And the symmetry case, we consider three cases. One is the case where both strains doesn't have the ability to go to quiescent. Another is the case where both of them have a small rate of going to quiescent. So this rho parameter of switching from active to quiescent state is smaller. And then you also look at a case where both the strains have a high rate of switching to quiescence. And here, when you change the transmission rate, you can see that for low transmission rate, the infection level of the system with no quiescent is much higher compared to the infection level of system with high quiescent. But it somehow vanishes as we increase the transmission rate. So what could be the reason? So we have some understanding of this. So we look at these two systems as separate cases. So we have a low transmission scenario and high transmission scenario. And then if you look at different level of quiescent rates and look at the infection level, it is consistent. So you see that when the transmission rate is low, as the quiescent rate increases, your infection level drops and the variance of the system also drops. But if you look at a high transmission level case, what happens is somewhat opposite behavior, but it's probably much milder. So I am not like 100% sure that this increase is a significant increase, but it does show a slight increase in the infection level. So what probably happening here is, so when you have low transmission rate, so if you look at the system, so here the inflow to these infected classes are smaller, and then if you push most of them to the quiescent state where they cannot infect new individuals, then somehow it kind of have a negative effect on the system. So if you have very less infected individuals and most of them go to quiescent state and they do not create new infections, your infection level drops. But in the other scenario, when you have very high transmission rate, probably it's a different case where you always get a strong inflow to the infected classes. So even if some of them go to the quiescent state, the ones there'll be something remain in the infected classes, they could still create new infections in the system. So probably that's why we could see this two different types of behavior of increase and decrease in infection when you change the transmission regime. Yeah.
That makes sense.
Yeah.
For low transmission rate.
Yeah.
Do you have thoughts on why that relationship changes in sign when you go to high transmission rate? So why does quiescence increase infections under conditions of high transmission? Why do infections go up with quiescence?
Yeah, means the only this is a bit, this is not that significant increase first of all, so probably it is more or less constant, so that is also possible. But since we try to extend the scale further to see that is there really a trend but then it still holds. I mean, even if it's the only explanation I have is, since this transmission rate is very high, you always push a lot of individuals to the infected class. So even if some of them go to the quiescent state, you still have something left in the infected class to create new infections and keep the infectant level higher in the population. But yeah, this has to be like evaluated more carefully, I guess. But yeah, so this is one thing we get from the symmetric rate. But the more interesting one is, of course, the asymmetric case when you have competition between two strains. So what happens there? Yeah, this is expected when you compete with a strain with quiescent with a strain without quiescent. The one without quiescent has higher level of infection, so they win. So the one with quiescent, of course, have a disadvantage in an environment where there is no fluctuations and anything. So the one without quiescent wins, the one with quiescent has a disadvantage. But the interesting result here is we try to look at the level of stochasticity in the system. So we plot the joint distribution of infected individuals in two scenarios, when you have one strain with quiescent in the system and when you have both strains with no quiescent in the system. And you can see that the variance of this joint distribution is quite different in two cases. So when you have quiescence in the system, you do have infections only by mainly by one pathogen, because the other one has a disadvantage. It's kind of kept in a very minimal level. Uh, the infections are mostly caused. So in this scenario, say pathogen 1 doesn't have quiescence and the pathogen 2 has quiescence. So all the infections for the one with quiescence is caused by pathogen 1. But so if you look at this contour plot, you can clearly see that when a system without quiescent, the variance or the stochasticity in the infection is quite higher compared to the one with quiescent. So which suggests that maintaining a strain with quiescent in the system still has an advantage in reducing the overall stochasticity of the system quite a lot. So this is been can be somehow relate to this moving average kind of effect. So this quiescent compartment in the system is giving some sort of a buffer in the system which behaves like a moving average, kind of averaging out the noise in the system to quite a lot and reducing the overall stochasticity in the level of infection quite a lot. So this is the main interesting result we have from this model. So with that, I probably will conclude the first part of my talk, which is about this model and the population level changes in the model. So with this model where we consider the quiescent strain, we show the existence of a polymorphic equilibrium with parasite dormancy. And the parasite dormancy seems to reduce the infection when the transmission rate is low. But for high transmission, the quiescent rate seems to generate a mild increase in infection level, but this has to be investigated more carefully, I guess. But the most interesting result is when there is competition between these two strains. So when there is difference in quiescent rate between the strain having the high quiescent rate seem to have a disadvantage and always kept in very small level in the population. But maintaining this strain has some advantage in reducing the overall stochasticity or noise in the system. So to having this strain with the property of quiescent kind of reduces the overall stochasticity of the system. So this is an interesting result. Okay, so now I will go to the other part of the talk where we wanted to look at the genomic signatures of host-parasite coevolution. So we have the same type of scenario. We have the host and the pathogen. And we want to look at the footprint of this interaction in the genome level. So this work is mostly done with Hannah and Lucas and Aurelion. Okay, so here it's not exactly the same model. So we are we are kind of thinking of connecting these two, but this is a slightly different model. So we still have two pathogen, but we also have two hosts now. So it's a slightly high dimensional system. So we haven't added the quiescent compartments to the system yet, but we could do it at some point. So this is the system we want to work with. We have a host 1 and host 2. So we have two host types and two pathogen types. So it will create something like 6 classes in the population. So you have host 1 infected by parasite 1, I call it as I11. Host 1 infected by parasite 2 is I12, and so on. Then here also you could also, I'm not going into the details of the model now because we are not really going into the population level changes of this model so much. But we can still write down the differential equation for the number change of all of these compartments based on the new birth and death and the infection rates, some equations like this. Okay, so here what we are interested in is the changes in the genomic level. Okay, so there is a host genome and there's a pathogen genome, and they are interacting. These two species are interacting. So what is happening in the genome? So here in the genomic level, do you expect that all part of the genome get equally affected by this interaction? No, right? So there could be some regions in the genome which are mainly the drivers of coevolution, and there could be, so there can be some other regions which are mildly influencing the process, and there could be many other positions in the genome which has nothing to do with the coevolution process as well. So there could be this neutral gene regions, and there could be this major genes which are mainly the drivers of coevolution, and there are many other genes which could be some sort of a polymorphic trait which is distributed along the genome, which can have milder effects as well. So the idea here is to somehow identify this genomic regions which drives the coevolutionary process. Okay, why we do it? So there's an interesting study. There are a few more, but I'm just showing one of them because this is the study we have the data from. So I'll try to compare it with our model setting as well at the end. So yeah, why we do it? Of course, it's important to identify these genomic regions which drive the coevolution because it can provide advantage in vaccine and drug design. So there is many studies where they try to identify which part of the human genome is associated strongly with which part of the pathogen genome. So here we have the human genome in the bottom, where they show different chromosomes in different numbers. And then you have the pathogen genome here. So this is the hepatitis C virus, some amino acid position, something. And then they draw the association level between all of these pieces of the genome of host to the pathogen. And then they somehow highlight the lines which have the maximum level of association. Interestingly, you can see that most of the high level of association from the human genome goes from somewhere in chromosome 6. And not so surprisingly, it happened to be the histocompatibility complex, which has to do something with the immune system. So this is the region in the human genome which is strongly associated with the some few parts of the pathogen genome. So probably it's more interesting to identify which part of the pathogen genome is really associated with the human genome. Okay, so what we try to do is a similar line, but we wanted to also look at the interspecies LD, which is like the association level of all these allelic variations in two position. It's a bit difficult to read, but basically, so if you have the host genome, like a plant or something like this, and the pathogen genome, so you sequence the whole infected host plant, and then you sequence the pathogen in that plant, and then you have, so which pathogen is there on which host. We know this information, assume that we know this information, and then we could actually compare the genome of both these species. So here we are looking at a biallelic position, which means, so I can call this C allele, the individuals which carry the C alleles as my host 1, and the individuals which carry the G alleles as my host 2. Similarly, in the pathogen, I can call individuals which carry the T alleles as pathogen 1, and individuals which carry the A alleles as a pathogen 2. And then I look at the association level between all the SNP pairs in the host and the pathogen genome. So here you can see that there's a strong association. So whenever I have a G in the host, I always have an A in the pathogen. Or whenever I have a C in the host, I always have a T in the pathogen. So this indicates there is a strong association between the host and pathogen genome. And we are trying to identify such positions. So for that, we have this parameter, which we call it as the infection matrix, so which is a 2x2 matrix for a system with 2 hosts and 2 pathogens. So you have this alpha matrix, so which is basically the main model parameter which we want to estimate at the end. So each entries of this infection matrix represent the probability of a successful infection when a host and parasite come together. So for example, so alpha 11 is basically giving us the probability that host 1 encounter a parasite 1 and so on. So what is the probability that it result in a successful infection? Okay, so let's try to understand this a little bit better. So you could have different infection metrics in the system. So we look at different cases, which is probably biologically interesting. So basically, you have a system where I have host plant 1, which are the blue plants, and host plant 2, which are the red plants, and parasite 1 is black dots, and parasite 2 is the gray dots. And then if I have infection metrics like this, say then there is one host. So the rows are the host and the columns are the parasite. So the first row is 0, meaning host 1 is resistant to both pathogens. So when I look at the population, what I would expect to see in this scenario is like all the host 1 plants will stay healthy and the host 2 plants will carry both pathogens. Similarly, for parasite infectivity, if I have a strong difference in parasite infectivity, where for this particular case, parasite 1 can infect host 1 and 2, and parasite 2 cannot infect any host. Then you expect to see something like this. Both hosts can get infected, but only by parasite 1, and so on. So you could actually design, so for a particular infection matrix, you expected to see very different types of phenotype in the population. Okay, so now we look at a realistic sample we might get from some field or something. So you could have 2 types of host plants and then you sequence them. And then you categorize them based on which host plant is associated with which pathogen. So this can be done. So the host 1 getting infected by pathogen 1, I call it as F11, and host 1 infected by parasite 2 is F12. And this is basically the healthy plant, which somehow has some resistance. Even if it's in the infection prevalence area, it's still healthy. It can happen if it's resistant to both the pathogen. So I group them as another class. And so the same thing. So then you could have another region in the field where the infection is not spreading. So there you also can sequence some healthy individuals from this region. So we have all these groups in the population where you somehow have the frequency of each of this group from the population. And then we try to design some measures which could capture the different scenarios or maximize for this different scenarios of interest. So we somehow come up with so this is basically the interspecies linkage disequilibrium kind of measure, which is a little bit familiar, but we also come up with similar measures which could actually capture different types of interactions or maximize for different types of interaction. And it's straightforward to assume that we can easily calculate these numbers from our sequence data. So if we have the infected host and associate parasite data, and also if you have the healthy host, I could actually calculate all these fractions I want based on these numbers. And then I can also obtain this values of all these different indices based on these fractions from the data. Yeah, so the interesting thing is like if I have information about the value of all these indices for each of the STIP pairs, I could actually say something about what could be the underlying interaction going on between these 2 STIP pairs. So so this is colored based on the behavior. So sometimes you expect to get a 0 for some index value, some positive value or negative value based on the expectation of this indices values from theoretical calculations. And then we can see that for each of these metrics of interest, you can actually uniquely identify what could be the underlying metrics based on this 4 index values. So this is the main idea. So now we try to apply it to this human HCV data. Okay, so what we have is, so we have this data from the human genome data and the HCV data. Lucas has done all this calculations of different, calculating the values of different indices we have from the data. So for each SNP pair in the host and the pathogen, we calculate the value of all of our 4 indices. And then this is already identified that some, so in the first study I showed, they have already picked some positions in the genome which actually shows the maximum level of association, right? So we already have this information from this study that there are few SNP pairs which show the maximum level of association. So can we recover this? So this is the first question from with our indices values. It seems like yes. So this is the neutral expectation which we get theoretically with a combination of 2 of our indices, and all the SNP pairs which they claim to be significant comes as an outliers for with yeah, I mean, so you can with the host type in the sense the human genome, you pick all the positions which have 2 alleles. So we really don't know. It's not really, yeah, it's some genetic differences you could identify in the genome for humans as well, right? So you have the polymorphic SNPs in the human genome. In the chromosome 6 also, you have some SNPs which does have 2 alleles segregating. Yeah, so it's not like everybody has exactly the same sequence there. Yeah, we somehow look at SNPs which have 2 allelic states, or we somehow come. Yeah, yeah. So this is a lot of data. So you could have like many comparisons of all the SNP pairs of the host and the pathogen. The host is like sequence of human from different regions, probably from Africa, from Europe, or from, and then you could actually identify. So when you assign all this genome of different human genome, uh, and I think they have some European samples and Asian samples and everything. So then you can identify some positions in the genome where you have 2 alleles segregating, and you pick all those positions and then you compare with the parasite genome to get this, to calculate this in these values. Okay. Yeah, so basically it seems to work. So with our indices, if the neutral expectation is this gray points, and all the SNPs which they claim to be having a significant level of association comes out as outliers with our indices also. So which is somewhat it convinced that our idea works. So we could actually identify the SNP pairs which show a significant level of association with these in these values. But what extra we could offer is to say something about the underlying interaction. So this is something which we could provide with our setting, that we can actually estimate this alpha metrics for each of the SNP pairs which show the maximum level of association. So for example, so from our setting, we would predict that the most significant SNP pairs of the 500 of them shows mostly this kind of an infection matrix, which suggests, so it's not exactly 0 1s. I'm just kind of, I mean, so this biologically you will never get a 0 1. It's anything between 0 and 1. You can only have some differences in one, or some slight preferences on one pathogen and another, but it's not like exactly 0 1s. But what we see is like, so there is a strong preference of pathogen 1 on host 2. So many of the individuals who carry the genotype of host 2 are getting infected by pathogen 1, but the other infections are a little bit milder. So it somehow indicates that there is some sort of a preference for this particular pathogen to infect individuals which have this genotype. So this is the extra information we could provide if we do this method. So we could, yeah. So basically, yeah, that's it. So here I tried to explain that we can actually get some information in the genomic level also about how coevolution influenced the genome of both the species. And here we managed to come up with 4 indices which can actually recover the information we get from co-jiva studies, where you compare the genome of, in a very empirical setup, when you compare the genome of a host and pathogen and identify this significant associations. But our method is a slight advantage, which can also say something about the underlying interaction, basically the interaction metrics or the alpha metrics. And we try to apply our model to this human HCV data and also recover the most associated SNP pairs which been predicted before. And we also have some predictions about what could be the prominent infection metrics which is being in this human HCV interaction. Okay, so with this, I would like to thank everyone who is involved in the work. Usman is here, and Aurelion and Johannes has done the first part of the work, and the second part of the work. Hannah was there as a PhD. Now she moved to US for a postdoc, and Lucas is here, and Aurelion will soon be here. Yeah, okay, thank you.